This is the microalgae physiology lab that is just, which has completed construction and is meant to study physiological responses of algae to the environment. There is no algae growing now because we are running some background checks in the system to make sure that everything runs well. But next week we are going to seed microalgae here and in all 4 bioreactors and start studying the physiological responses to changes in temperature, changes in salinity, in nutrients, in the supply of CO2, the light levels, and then get all these responses, which are the basic parameters controlling or affecting the production of microalgae. With all these responses, we'll then integrate all these responses into a simulation model in order to develop a tool to use them in the management of larger outdoor systems. Okay? And we're going to be looking at different strains. There are different strains of microalgae that have the potential to produce different amounts of lipids. Obviously, we're going to be looking at microalgae that has a high capacity of production of lipids, because lipids are then extracted to produce and refine into diesel or biofuels. Along with that, after the extraction of lipids from the biomass of algae, the microalgae, there is a residue that we are going to analyze for its quality to be used as feed for animals, either shrimp, fish, or cattle. And eventually the possibility of using these residues of microalgae as soil fertilizers. So we are just starting with this project, and we are working in very close coordination with the Mariculture Lab in Flower Bluff, directed by Dr. Zahi Samoa, the Mariculture Lab in Port Aransas, directed by Dr. Addison Lawrence, and other folks that are participating in this project.